These are the lecture notes that modern understanding is built on. This is more modern, going to be more. So modern genetics free published work at a paper titled and also for um, so the work was kind of converted that year work and that is when after understanding that log, log, um, scientists biologists would, what DNA um, Mendel in his time he had no idea he had no idea about okay um, so they tried using a light wouldn't wouldn't be helpful under a light but as microscope observed so that you know cell division you know individual their banding pattern or some of non but today we know so we know that it was discovered after Gregor were responsible for our and then after that it was so the chromosomal takes the chromosome so genes carried on okay um, and to further explain this as I so chromosome inventory terminate that sorting of chromosomes winds up so that half with half chromosome um, parent double copy even though male differ in size they have the same number of chromosomes. So this suggests that um, to inherit instead of child, they don't always. Now the chromosomes were to produce first a zygote that also um, zygote the cell after the egg egg fertilization 46, and then that zygote divides cell all of the body chromosomes. Okay, so these scientists are usually easier plants. They weren't complete they were they were studying the genetics, the inheritance, and each year that they actually take anaphase, anaphase. Okay, so Eleanor Carruthers demonstrated um, of chromosomes in grasshoppers. Uh, so that was for such, and then every sex chromosome was actually based on work done on fruit flies, Drosophila melanogaster. Drosophila are fruit flies, and there's a lot of genetics experiments that have been done on fruit flies. They have been really helpful in understanding heredity, even applying it to human heredity. So in fact, here's a fruit fly all blown up for you. With red eyes, fruit flies can have various colors of eyes. They can have red eyes, white eyes, and then they can have different shades of brown eyes. But this is the karyotype. A karyotype is just an image of the chromosome sets of an individual. So this is the karyotype for a fruit fly. It looks like either 3 or 4 sets of chromosomes. I can't tell in here. That doesn't look like chromosomes. So this is a karyotype of a fruit fly that contains 3 sets of chromosomes, so a total of 6. Just a review, that means that the diploid number for the Drosophila would be 6. Diploid or 2N number would be 6. And the haploid number, the number of chromosomes found in the egg and the sperm, would be 3. Okay, and a human karyotype is shown on the right, and you can tell that this is the karyotype of a male because the 23rd pair of chromosomes contains an X chromosome and a Y chromosome. And this is also a good time to talk about what a Y chromosome actually is. It is not shaped like a Y, as you can see here. The Y chromosome is actually a small piece, or like the bottom half or the top half of an X chromosome. It's just an X chromosome with some parts of it missing. So it does not contain as many genes as the X chromosome does. So the X chromosome is necessary in males and females because it supplies the missing genes. But males that have a Y chromosome don't go without genes. It doesn't mean they have less genes or anything like that. It just means that they're going to express whatever genes are located on this part of that X, that they're going to express those genes. They won't be heterozygous. They won't be able to be carriers. They'll only have one copy of the genes that are located on this part that I've circled, the part that's not included in the Y chromosome. But the point is here, this is a karyotype that shows all the homologous pairs, all the 23 homologous pairs for human, and we can tell it is a male because there's an X and a Y. The presence of the Y makes it a male. Now, sex linkage was the first demonstration that genes were on chromosomes. And we talked about sex-linked traits, and we talked about the fact that eye color in fruit flies is sex-linked. So, in other words, the gene for eye color is located only on the X chromosome. And this concept of linkage was used to map genes. So we have now mapped all of the human genes on all of the chromosomes. But that was the Human Genome Project, which has been completed. It was completed a few years ago. But they began to use this concept of linkage to start mapping the chromosomes. In other words, to show the location of every gene for every trait. Okay? So Morgan's work with sex linkage showed that eye color, the trait for sex, and the trait for eye color were inherited together, that they did not segregate independently because they were linked. So anytime you talk about linkage, anytime there are traits that are linked, they violate the law of independent assortment. They do not assort. Most traits that are on different chromosomes do follow the law of independent assortment, the second law. They do randomly assort or randomly sort themselves in metaphase, randomly line up in metaphase in different positions. So in other words, the sperm and the egg do get a random assortment of traits because the traits segregate independently when they're on separate chromosomes. But if traits are on the same chromosome, it doesn't always work that way. They don't always segregate independently when they're located on the same chromosome. The only time that they're going to is if a high percentage of crossing over occurs of those traits. So traits that are closer together on a chromosome tend to be found together in the offspring, in the organism. And if the traits are closer together, they segregate less often than you would predict in independent assortment. So the closer together that traits are on a chromosome, the less crossing over there's going to be. The farther apart that they are, the more crossing over that you will see. Crossing over will happen more frequently. Now, this is an image that shows inheritance patterns of unlinked and linked genes in fruit flies. And I'm going to stop here and we'll start in part 2 with this slide, which is slide 12.